will you be coming back, or should I? Okay. Great. Hello. It's not really spring out there. It's just sort of in between. Let's do a sound check. Can you hear me back row? Can you hear me in the back row there? Okay. Has anybody tried to use the new YouTube video? Does that work as well as the Google video does? Okay. Some people have asked me about downloading, and I don't think YouTube includes a link for downloading, but I know that Firefox has add-ons that allow you to download YouTube videos, and I would imagine there are others as well. As I think I mentioned, Google is in the process of shutting down video for plain old people like me, and shifting people over to YouTube. And it was clear that there was starting to be some degradation on the YouTube videos, so that's why the switch. Any sort of general course-type questions before we talk biochemistry? I'd like to make a reminder that we have another hour exam coming up. A week from today will be the third hour exam. And come on, open. There we go. The third hour exam will be a week from today. It will be through Chapter 16, which is the integration of metabolism. I'll send around a little notice about who should go where. There will be a review session again. That'll be on Thursday at 6 p.m. in this room. So have that in mind. Any other questions before we get started? Okay. Let's see. We were in, okay, somewhere around here. We're on. Okay. Well, I think we were here. Is that right? Oh, thank you. That's important. Thank you. And the lights. Okay, let's try that. Are things working now? Okay. Is that too dark? All right. So we talked on Friday about the importance of nitrogen, the fact that gaseous nitrogen N2 is extremely expensive energetically, and some of the ways in which organisms have come up with fixing it and then moving it around between different amino acids and so on. But life does not run on just amino acids, although proteins made of amino acids accomplish 95% of the catalysis. Nucleotides also contain nitrogen, and therefore nucleotide biosynthesis and degradation is extremely important. Without it, there cannot be continued life. Fortunately, there are only 5 nucleotides. Some of the nucleotides that we have are synthesized from precursors that include amino acids. We are capable to some extent of taking in nucleotides from our diet, and some of them are simply rearranged or moved around from pre-existing nucleotides. Now, the purines, which are the adenosine and guanosine, the big ones, if you like to think of it that way, the ones that are fused rings, both AMP and GMP are built onto a base of something called, for short, PRPP. And it's called PRPP because its full name is phosphoribosyl pyrophosphate. So you now know why we call it PRPP for short. And PRPP starts, as you might expect, from ribose 5-phosphate. It costs 2 high energy bonds coming off of ATP. So here we have the kind of foundation onto which we're going to build the purine nucleotides. And the enzyme is called, pretty rationally, ribose phosphate pyrophosphokinase. So it's the enzyme that adds pyrophosphate onto ribose phosphate. Purine synthesis itself is fairly intricate. There are 10 steps in it. We're not going to go through each of those 10 steps because this is a one-semester course, not a two-semester course. But interestingly, there are 5 different molecules that give rise to different parts of the purine molecule. And let's take a little look at the backbone, at the structure of inosine. This is the first purine that's produced by the pathway. And when we walk around the ring structure, we can see that there are 2 nitrogens that have come in from glutamine. And glutamine, of course, was a nitrogen carrier, very important for amino acid synthesis. Another nitrogen from aspartate, a 4th nitrogen from glycine. So 3 different amino acids contribute to the ring structure of purines. A couple of carbons, 1 carbon here and another carbon there, that are coming from formal tetrahydrofolate. We mentioned when we were talking about amino acid synthesis the importance of tetrahydrofolate for single carbon additions and subtractions. So here we see again 2 very important tetrahydrofolate contributions, and 1 carbon coming in from bicarbonate. The rest of the carbons in the chain actually came in along with glycine, so you can see it there. So all of the nitrogens come from amino acids. The carbon backbones come from a variety of sources. So here we have IMP. Here's the inosine rings and the attached to the ribose 5-phosphate. So here's inosine monophosphate. And IMP gives rise to both AMP and GMP very easily. It doesn't take very many steps to do that. So if we start from IMP and we want to end up at AMP, adenosine monophosphate, we can attach an aspartate at the expense of a GTP, so it's kind of flapping in the breeze here, and split off the 4-carbon backbone, and that leaves behind the amino group we need to make this be AMP. So 2 steps from IMP, we get AMP here. It's just as easy to get down to GMP, slightly different steps. The first step, we're going to oxidize it. So here we're coming in with NAD plus and coming out as NADH. So we have oxidized this carbon. In the next step, we're going to take a nitrogen from glutamine and replace the carbonyl carbon to make an amine there. So both cases, we're using up nucleotide triphosphate. In the pathway down to AMP, we used GTP. And in the pathway down to GMP, we used ATP. And this is extremely clever. This is extremely clever. It allows for a reciprocal regulation so that you balance the production of GMP and AMP without having to have any very separate kind of mechanism involved. If you're short of GMP, you're not going to be making more AMP, and so on. So this reciprocal contribution helps provide a balance of the right nucleotides afterwards. And then both of them also use important and common amino acids. So here's aspartate there and glutamine there. So the abundance of amino acids will themselves, by just what they are, be part of the signal about whether things are flush and we're ready to make more stuff and therefore need more nucleotides, or if it's kind of lean and we're not going to go ahead quite so fast. So very clever connections simply by what compounds are used to generate the 2 pathways. So 1 common source, IMP, for both purine nucleotides, using both amino acids and nucleotides to balance the production of both. Pyrimidines are a little different. In the case of pyrimidines, we start by making the base first, that is the heterocyclic ring. And here you see the 6-membered heterocyclic ring on uridine. And again, we're going to be taking parts of amino acids. So nitrogen, again, from glutamine, and essentially the entire molecule of aspartate provides most of the rest of it, and again, a molecule of bicarbonate. So after this base has been made, it can be attached to PRPP. You remember we made PRPP as the sort of foundation stone for purine synthesis. For pyrimidines, we make the little ring and then smush it onto a PRPP. And the synthesis of this is inhibited by their products. So any of the uridine nucleotides, monophosphate, diphosphate, triphosphate, all three of them will inhibit the production of UMP. So there's feedback. And it'll be activated by ATP. And this is another example of trying to keep the balance of the mix of nucleotides about right. If you're accumulating pyrimidines, you shouldn't be making more of them. If you're accumulating a lot of purines, maybe you need some pyrimidines. So how to make CTP? UTP, of course, is useful. We need UTP for RNA. We also need CTP for RNA. And a very simple step, CTP synthetase. Glutamine comes in again. This costs us energy, ATP. And what was a carbonyl oxygen carbon in the pyrimidine ring here has now an amine group on it. But it cost us a little bit of energy. So there's CTP. But again, glutamine is involved. Now, what we've made so far is UMP and CTP, which are both ribonucleotides. Ribonucleotides are going to be used for RNA, ribonucleic acid. If you want to make DNA, you need deoxyribonucleic acids. You've got to get rid of that oxygen, right? And this is looking at the process of ribonucleotide reductase. Here's your ribose ring. Here's the 3' hydroxyl group that's going to be involved in making the polymer of a nucleic acid. Here's the 2' that is not involved in making the polymer. That's where you differ between DNA and RNA. And ribonucleotide reductase has almost like a Velcro stretch in it of sulfhydryl groups. It starts off with 2 that are sulfhydryls and 1 that's a radical. That radical is able to react and detach a hydrogen there. And then in that process, the sulfhydryl is restored there. The intermediate sulfhydryl is able to donate a proton to protonate that alcohol, which is kind of a surprising kind of a group. So now you have a protonated oxygen. And then this group, as you might imagine, can leave as a water molecule. And when it leaves as a water molecule, it leaves here a cation, what's called a carbocation. You wouldn't want to leave this thing like that. It's got a radical there and a carbocation there. That is not a happy molecule. And so the remaining sulfhydryl groups that are present on the enzyme, this hydrogen can be donated to neutralize that carbocation. You still have a radical there, though, so you wouldn't want to leave it like that. But if you remember, we had a radical in that top position at the beginning of the whole process so that the top sulfhydryl can transfer its hydrogen and restore that completely. That leaves the enzyme in this reduced oxidized state. This is oxidized here, and it needs to be reduced back down at the expense of thioredoxin. And the product of then is a deoxynucleotide diphosphate. Excuse me. Okay. All right. So, intricate and lovely. So now we have DUMP. I've always liked the name of DUMP. Deoxy-UMP, deoxyuridyl monophosphate. But we don't use DUMP for anything. We use UMP in RNAs. We use something else for DNA. And so in order to make something we can actually use to make more DNA molecules, we're going to have to methylate the pyrimidine ring. And the donor is again going to be tetrahydrofolate, in this case a methylene tetrahydrofolate. And the enzyme involved is thymidylate synthase. And by adding that carbon here, we can turn DUMP into DTMP. So now we have deoxythymidine monophosphate. And this will now be the correct base for participating in DNA synthesis. you absolutely have to have thymidine to make more DNA. And this enzyme, thymidylate synthase, is used only to make thymidine. So this is a crucial spot if you want to inhibit DNA synthesis. Inhibiting this enzyme will inhibit DNA synthesis. And methotrexate acts because it inhibits the restoration of the tetrahydrofolate. As you can imagine, that means methotrexate probably has side effects as well, but it does act as an anti-proliferative agent. This is the structure of the molecule, thymidylate synthase, and what you can see is there's kind of this is the electrostatic charge on the surface, and so it's got different charges at the ends and almost like a little highway in the middle that the compound can slide along during the process of finishing the reaction. All right, so far we've talked about these things as if all you ever did was synthesize things, but we also break them down. Amino acids are broken down for a number of reasons. They can be used as fuels. If there's an excess of nitrogen in the diet, the excess will be used as fuels. If there's a shortage of calories, proteins may be broken down to amino acids, which are then needed for fuels. And interestingly, the small intestine, by preference, likes to use amino acids as its fuel. Maybe that reflects the fact that it gets first dibs on things. In the liver, we see the reactions needed to use amino acids either as fuel or to use the carbon backbones from them to make glucose or ketones. If you are fasting, if you are not taking in calories by mouth or any other way, at some point, we'll see later on at what point, you will start breaking down your own muscles in order to provide the carbon backbones for gluconeogenesis. Now, what amino acids are used for are going to depend a bit on the structure of their carbon backbones. And so we describe amino acids as glucogenic, ketogenic, and a few or both. So we call an amino acid glucogenic if its carbons can end up in glucose, and we'll see the structural features that tend to be consistent with that. We call them ketogenic if they are used for fuels in the form of ketone bodies. And there are a couple of them that can actually make some of each. So let's take a look. Here we see the structure of asparagine. This is the amide down here. Asparaginase. Now we're assuming that we're trying to break down amino acids. We are either flush with them because we've eaten a steak or we're fasting. So asparaginase can cut off that nitrogen and release it as ammonia, consuming some water in the process, and now we have aspartate. Aspartate can transfer its nitrogen to an alpha-ketoglutarate to make glutamate, and that gives us now the carbon backbone for oxaloacetate. And you all remember that oxaloacetate is a precursor for gluconeogenesis. The logic here is fairly similar for glutamine. Glutamine is just one carbon longer. It can cast off its nitrogen, its amide nitrogen as ammonia, making glutamate. And the nitrogen on that can be transferred to something else, depending. And the carbon backbone released in that process is alpha-ketoglutarate. So, serine and cysteine. Here we see serine, and here we see cysteine. They're both 3 carbon backbone sugars. Serine, the alcohol, cysteine, the sulfhydryl. They both have 3 carbon backbones. They both give rise to pyruvate, which is itself a 3 carbon backbone, methyl group at one end, carboxylic acid at the other, carbonyl in the middle. And I'm mentioning that because there were people who didn't know how many oxygens pyruvate had. When we do this, if we're going to burn serine, its nitrogen here comes off as ammonia. If we're going to burn cysteine, we get both an ammonia off of it and a hydrogen sulfide. Pyruvate, of course, is a 3-carbon fragment that can be used for gluconeogenesis. I know this looks a little funny, but otherwise it all would take up a lot more space. So. This is sort of a summary of some of the places that different things end up, sort of help you see how they get there. So asparagine and aspartate are 4 carbon sugars coming to oxaloacetate, gluconeogenesis, and glucose. Serine and cysteine are 3 carbons to pyruvate. By adding a carbon dioxide at the expense of energy, of course, pyruvate carboxylase costs energy. We get oxaloacetate. Out of the citric acid cycle, we can withdraw alpha-ketoglutarate and make or rather, we can feed things into the citric acid cycle and draw out 4 of the carbons as oxaloacetate. So this explains why these amino acids, 1, 2, 3, 4, 5, 6. So these are there are 6 amino acids here that are all of them very clearly glucogenic amino acids. They can make glucose. Threonine's a little trickier. It's one of the ones that I mentioned could do both. So threonine has 4 carbons. It's a branch, has an alcohol branching sort of partway down. It can be oxidized, giving you an NADH that you could use for energy. And now you have alpha amino ketobutyrate. So there's a carbonyl oxygen there. And a CoA molecule can come in and pick up that 2-carbon fragment. So that 2-carbon fragment is now acetyl-CoA. And the other 2-carbon fragment is glycine. So this part could be ketogenic. The glycine can be glucogenic. So what happens to the glycine? Well, you may remember that some glycine can go to serine, in which case it would end up in pyruvate. But more of the glycine, when it's being broken down, uses something called the glycine cleavage system. And if we look at the glycine cleavage system, here's your glycine. It's a carboxylic acid amine or a single carbon. Glycine cleavage system can oxidize this, producing an NADH and tetrahydrofolate, carbon dioxide, and ammonia. So we have another molecule of ammonia we need to get rid of. We've been doing ammonia all along, so kind of mentally keep books on that. We've got to get rid of that ammonia sometime. But we also have a methylene tetrahydrofolate available for other purposes. So glycine could go several different places. Valine is a little strange. So here you have your valine molecule. It's got this branched chain in here. Remember, this is the branched backbone. It can transfer its ammonia, its amine group, rather. Its amine group to alpha-ketoglutarate, making a glutamate. And now what you have is a branched alpha-keto acid. And there is a specific enzyme called branched-chain alpha-keto acid dehydrogenase, which is a long thing, that does what pyruvate dehydrogenase does. That is, it takes branched-chain alpha-keto acids and makes CoA compounds of it. And so in the process, we can oxidize it a little further. You can see here that we've lost one of the carbons as carbon dioxide. This is giving us still a branched CoA compound. And this then can be further oxidized through a acyl-CoA dehydrogenase. And that creates a double bond there. And with other rearrangements, we can end up with a succinyl-CoA. This is a complicated process, and it's an essential process. It was first really understood through the detection of a human genetic disorder called maple syrup urine disease. Sorry. It's called maple syrup urine disease because a Norwegian mother noticed that her 2 sons, who were developmentally challenged, had urine that smelled like maple syrup. And she kept nagging pediatricians until someone figured out what was going on. So, aromatic amino acids are even trickier to get rid of, as you can imagine, with all that stuff in there. The aromatic amino acids are another example of amino acids that are both glucogenic and ketogenic. They can do some of each. So if we start with the phenylalanine here, for example, we actually have to make it into tyrosine to get rid of it. A phenyl ring is not a very reactive ring. And so if whether we're making tyrosine in order to make more amino acids, or if we're making tyrosine to get rid of the excess phenylalanine, this step is needed either way. So phenylalanine hydroxylase can hydroxylate it right there. An enzyme called transaminase is going to again transfer the amine group over to probably oxaloacetate or alpha-ketoglutarate, something like that. And now we have an alpha-keto acid. This is para-hydroxy phenylbutyrate. You can see that that looks kind of like phenylpyruvate. Phenylpyruvate. That looks like pyruvate, doesn't it? And then a dioxygenase comes in. Excuse me. A dioxygenase comes in and oxidizes that ring some more. So now it has two hydroxyl groups. And this compound is called homogentisate. The final step in its degradation is going to require another oxygenation. You can see here additional carbons that have been oxidized. And this then is going to be able to break down further into acetoacetate and either alanine or fumarate, depending on whether where you were starting from. This enzyme is the enzyme that is missing in phenylketonuria. Phenylketonuria is one of the commoner genetic disorders, and in all states of America now, it's necessary to test newborn infants for the presence of the enzyme, because if this deficiency is detected within days of birth, a child can lead a fairly normal life. But if it's not caught early, it can lead to gross accumulations of phenylalanine in the blood and generally bad effects. A second important step in terms of human genetics is this step. If this enzyme is missing, a disease called alkaptonuria occurs, and this is excessive, and the urine turns black, which must be very distracting. But both of them can be handled through diet. And neither of them are a problem in utero, because, of course, in utero, the mother is handling all the metabolism, dirty work for the infant. So we've been merrily cutting ammonias off amino acids left, right, and center as if there were no tomorrow. We cannot just let that happen. At least we can't. Ammonia is, for us, toxic. A bit of ammonia in the kidneys is needed. If you're going to excrete an acid, ammonia makes a good counterion, but that doesn't come close to taking care of the total needs. We're going to see later why it can inhibit the citric acid cycle if you have too much ammonia around. So our solution for that is to make a molecule called urea. You can see in this molecule that it can carry off 2 nitrogens for every carbon. Urea is extremely soluble, and it's non-toxic under any concentration that a living organism can make. Probably 80% of the nitrogen that's disposed of is taken off as urea from the liver. So where do we get the nitrogens for that? Okay. You noticed a couple of times we transferred nitrogens onto alpha-ketoglutarate to make a glutamate. It's like, well, what do we do with that? That we can take care of with a mitochondrial enzyme that can release that amine group from the glutamate. And intriguingly, this mitochondrial enzyme can use either NADP or NADP plus or NAD plus. It does not care which of the two dinucleotides it's going to use. One of the very few enzymes that does not care. And so if you look at the carbon to which that amine group is attached, the consequence of this enzyme is to further oxidize it, and then by replacing that with water, you end up with an alpha keto acid. So this is a way to remove ammonia and leave behind alpha ketoglutarate. These reactions are reversible. So to some extent, it could be used to fix ammonia to the extent that the concentrations in a cell and so forth are permissive of that. So we have, we've made ammonia. We've got lots of ammonia from one thing or another. Ammonia by itself is not going to be very usable to attach to things. The energetics are still not very favorable. So we have to make, in effect, an activated form of nitrogen. And that's going to be, that activated form is going to be something called carbamyl phosphate. So here's the nitrogen that we're trying to get rid of. And this is going to be the activated intermediate we're going to use in the next step. So you might wonder how we got there. And it's going to be somewhat energy intensive. You start off with a bicarbonate ion, an ATP, and you, at the cost of one high energy bond, you now have carbonyl phosphate. So if this is a carbonyl phosphate here, and this compound is one with which the ammonia can interact, and it attacks in here. That gets rid of the phosphate. We now have carbamate. So carbamate is getting on towards looking like urea, but not quite. It still has an oxygen over here. And this carbamate is going to be further activated by another ATP. And so now we have carbamyl phosphate. So the nitrogen there in an amide and the phosphate on the other end. Carbamyl phosphate. So carbamyl phosphate is the form in which nitrogen enters the urea cycle. The urea cycle is the pathway in the mitochondrion through which excess nitrogens can be turned into this nice, non-toxic, nitrogen-dense molecule, urea. So I'm going to give you a bit of an overview of the pathway, and then we'll look at some of the reactions in a little more detail. Here's our carbamyl phosphate. It's an activated form of nitrogen. And we're going to start with carbamyl phosphate and an amino acid. If you look up there, that's an amino acid called ornithine. And ornithine, if you look at it, looks kind of like lysine, except it's missing a carbon. And if you notice the ornith root in there and you think maybe ornithine was isolated from bird poop, you probably wouldn't be too far off. Anyhow, ornithine is going to be fused with the carbamyl phosphate. We're going to smush that onto the amine group there. And now we have citrulline with the amide group down there. The next step in that is going to be taking in a molecule of aspartate at the cost of ATP and actually adding the aspartate on the other side of that carbon where we had the carbonyl bond there. So now we have added entire aspartate backbone there, which we're immediately going to throw away as fumarate. So the carbons of aspartate came off as fumarate and left the nitrogen behind. This was expensive. It cost us pyrophosphate. But when the fumarate comes off of this molecule, which is called argininosuccinate, it leaves behind, not surprisingly, an arginine molecule. This arginine is just plain old arginine. This arginine is the amino acid that we use for protein synthesis. So this is a case where arginine is used as an intermediate in a cycle, as well as our one we're used to thinking of it as, where it serves to make proteins. But this guanidino group down here at the end was always kind of strange, right? This 3 nitrogen cluster around the carbon is very resonant, very, very, very basic. By adding a water across that, that splits off a urea, and it reconstitutes the ornithine that we started with. So you can see how this could be a cyclic reaction. Every time you feed in a carbamyl phosphate at the top and an aspartate at the side, you can draw off a urea molecule and do it all over again. And this is just letting you see those reactions in a little higher detail. Carbamyl phosphate again, nitrogen, carbonyl oxygen, phosphate, citrulline down here with its amide group. Again, this is an expensive reaction costing us a pyrophosphate. So So the urea cycle as a cycle costs 2 high-energy bonds, but making the carbonyl phosphate, of course, costs energy as well. Here's the fumarate coming off. We can see there's been some reduction going on there. There's the arginine. So this carbon backbone is entirely the carbons of aspartate. The actual splitting reaction is free. It just takes an enzyme called arginase that can hydrolyze this bond to liberate the urea. So the oxygen is obviously getting added onto that carbon and regenerates the ornithine for the next cycle. People have sometimes joked about Krebs' bicycle. Hans Krebs discovered the citric acid cycle, which you may also hear of referred to as the Krebs cycle because he was so prominent in it. But Krebs also figured out the urea cycle. And since the urea cycle and the Krebs cycle, or citric acid cycle, are joined, biochemists can joke about Krebs' bicycle. Biochemical jokes are not going to make it onto Jon Stewart. But what can I say? But here, this is just to kind of emphasize for you the intimacy of that relationship. Here's fumarate, goes to malate, goes to oxaloacetate. Those are all reactions of the citric acid cycle. Urea cycle and citric acid cycle are both right there in the matrix of the mitochondrion, so they're right there to do it together. Now that you've got an oxaloacetate regenerated, transaminase can take another nitrogen off to make another aspartate. So this aspartate is just acting as the carrier of nitrogens into the urea cycle. And so this kind of tries to synthesize for you the different ways in which amino acids are functioning their nitrogens into the into the Krebs cycle, into the urea cycle, excuse me. The urea cycle is pretty expensive. It costs you a number of high-energy bonds between the carbonyl phosphate and the aspartate being added on. It costs a number of high-energy bonds all around. So you don't want to have it running unless you need to have it running. If you're breaking down a lot of amino acids, you're probably going to have a higher concentration of amino acids than at rest, say. And this gives rise to a molecule called N-acetylglutamate. And this is kind of a cute way to regulate the related pathway. So here's your glutamate, and acetyl-CoA, which would come, for instance, from burning fat, can make you an N-acetyl. If the acetyl group is on the nitrogen, N-acetylglutamate. And this N-acetylglutamate is an allosteric regulator of carbonyl phosphate synthetase. Since carbonyl phosphate synthetase is the committed step for the urea cycle, you can increase the flux through the pathway by increasing the activity of that enzyme. So this is a way of coupling the activity of the Krebs, a lot of cycles. This is a way of coupling the activity of the urea cycle to the abundance of amino acids being broken down, and also coupling it to some extent to the availability of energy in the form of acetyl-CoA. So urea is our principal way of getting rid of nitrogen when we have an excess. But there are other approaches to the problem of dealing with extra nitrogen. Both birds and reptiles are ecologically so situated that they need to conserve water. Either they live in dry environments, or they have to keep their body mass down, or a variety of other reasons. So they have a slightly different solution, and they make something called uric acid. Here's what uric acid looks like. Uric acid is not very water soluble. It tends to precipitate. And so the excretion of these animals is simply sort of to dump this in little white piles on your car. Well, if you've parked near the Old Capitol Mall, they do. And this is what that looks like. You have xanthine, which is obviously a purine core structure. Xanthine oxidase oxidizes it further, and you end up with this nicely oxidized ring structure that's water insoluble. Uric acid is also one of the byproducts of nucleotide degradation. Now, in general, we don't have a vast excess of nucleotides, but if someone has a very rich diet, they may have an excess of nucleotides. And in individuals who are suffering from that, you can end up with gout or kidney stones. That is, if uric acid comes out of solution in your toe joints, you're going to have gout. If it comes out of solution in your kidneys, you'll have kidney stones. So the urea we make goes out into the world. Like most organic things, it doesn't stay there forever. And there's an interesting enzyme called urease that can take urea and add water and recover the ammonia. So urease is a very cheap way for fungi and for bacteria to get nitrogen back out of urea. And then they can fix that ammonia however they like. Urease is interesting because it was the first protein to be crystallized. So it was the first thing to prove that proteins were a thing and not just a mush of stuff. And fish have the advantage that they live in water, so fish simply excrete ammonia. They don't need to bother with all of this biochemistry. So, and actually, that's the last of that. Do people have questions about nitrogen metabolism, nucleotide synthesis, the urea cycle? Is that a, no, okay.